All right, welcome everybody. So before I introduce today's speaker, do we have any announcements, any burning things people need to know about? Cool. All right. Well, today it is my great pleasure to introduce Chris Junium, who I have known for, gosh, at least 10 years, because we go to the same Gordon conference, which is kind of like science camp. And it's about organic geochemistry, which many of us will recognize as being slightly niche, but it's a lot of fun. Anyway, so Chris is coming to us from Syracuse University. Before that, he was an Agaron Fellow, and before that, he got a PhD at Penn State. And he is best described as a geochemist, isotopist. He's a little agnostic as to which element he thinks about isotopes in. And I don't have a fun fact. No, I do have a fun fact. Labware can be taken home and used for cooking. Foodware should not be taken into the lab. All right, take it away, Chris.
I should say lab techniques can come home for cooking, but you know, the centrifuge is a wonderful kitchen device. Well, thank you so much, Hillary, for inviting me and introducing me. It's a great pleasure to be here and to get a chance to talk to you a little bit about some things hopefully that you're interested in, some things that I'm interested in. So today I'm going to be talking about Earth's nitrogen cycle in a couple of different contexts as a way to try to understand the history of Earth's oxygenation. And I just want to call out, at least here, I have a mess of wonderful collaborators with whom I've done a lot of this work, but particularly a former PhD student, Ben Uvegas, is threaded throughout this talk and highlighting a lot of the work that he did in my lab as a PhD student, and then he keeps coming back to my lab to do analyses, which is great. And then right now a lot going on with Phoebe Cohen's at Williams College. All right, so let's get going. This image that you see here is of an oceanic anoxic event. This is carbonate-rich chalk here and organic matter-rich, we would call a black shale. And we go from basically no organic matter to 10 to sometimes in some cases up to 30 weight% organic carbon. And I've been infatuated with these events since I spent 2 months on an expedition with the ocean drilling program back in 2004 as a master's student. And I haven't really let them go. And they've informed kind of the rest of my career in the way that I think about, especially nutrient cycling and nitrogen and a bunch of other stuff as well. So when we look at this kind of picture, we recognize that there's a lot of change going on. And then if you think about the world in terms of nutrients, you have to understand that something has changed with respect to the input of nutrients into the system that has allowed organic matter to increase in this rock. And then also matching changes in the redox state of the ocean, thus, oceanic anoxic events, and we'll get into a little bit of the details of that. So first, when we're thinking about nitrogen, right, it's one of the most important limiting nutrients that we have in the ocean and land for all productivity. Most of it on Earth, however, is present as the very stable form of N2. If we think about blooms, in this case of calcareous nanoplankton, very important primary producers in the surface ocean. They use a form of nitrogen that's primarily in its oxidized form, nitrate, and they reduce it to ammonium that is at the proper redox state that they can put into their proteins. Now, they don't do this themselves, right? So they have to get it from somewhere. And in this case, the enzyme nitrogenase is the key component for mediating this transition, and it's primarily used in the modern ocean by cyanobacteria. There are other organisms that have nitrogenase. It's found all over the place, in part because it's a really, really useful trick to have. But this adaptation comes at a lot of cost, especially it requires a lot of iron to make this enzyme. It takes a lot of ATP to run this reaction. And it is also highly sensitive to the presence of oxygen. And if we think that most of the organisms that are capable of housing and producing this enzyme and fixing nitrogen from N2 are phototrophs that are performing oxygenic photosynthesis. You can see like you don't want to mix these two things together. And they've come up with some really novel and interesting adaptations to be able to deal with that. Here we have a filamentous cyanobacteria nostoc, a common one that is responsible for things like harmful algal blooms, build these special cells called heterocysts where they perform nitrogen fixation and then they pass that nitrogen off to their vegetative cells where they're performing photosynthesis. And then you have an organism like Trichodesmium, which is really important in the surface ocean, has other ways to handle that by doing it temporally or spatially, but not by making individual cells that where nitrogen fixation occurs. So if we think about the history of oxygen on Earth that's shown in this sort of generalized diagram, we have concentration of oxygen in the atmosphere. We have time down here going from basically the beginning of Earth history, 4.5 billion years ago, and then onward. We have a few important stepwise changes in the concentration of oxygen in the atmosphere. The first one coming from the beginnings of oxygenic photosynthesis, driving a transitional period that we kind of broadly call the Great Oxygenation Event. It maybe was this sort of old idea that it was this like snap of the fingers and oxygen increased, and now we recognize that this process extended over probably a long period of time. It was spatial, it had pulses, and we can begin to tease this out of the geochemical record. Beyond that, then, we begin to see evolution of different forms of life that maybe are adapting to oxygenic conditions. So the beginnings of eukaryotes, mitochondria and needs for, the oxygen demand of respiration. And then a little bit later on in the sort of the end of the Proterozoic and the beginning of the sort of visible fossil record, we begin to see metazoan life, which has generally a relatively high oxygen demand. And if we look at the geochemical record, we can trace this out really nicely. I'm not going to get into the details of what the mass independent fractionation of sulfur isotopes mean, other than that in an atmosphere without oxygen, we have these broad dispersion of values, and then once there is oxygen, that signal is erased. I'm sure that maybe you have students here and others who've heard about this before from certain faculty members here who have thought a lot about this. All right, so if then we think about the effect of oxygen on the availability of nutrients. So if you're adding oxygen to an otherwise anoxic system, the nitrogen cycle is highly dependent upon and responds to the presence of oxygen in a way that drives a lot of recycling. And in some cases, the important piece is that the redox cycling results in loss of nitrogen through the oxidation and subsequent re-reduction of that by microbes that are using it for energy. And so denitrification or nitrate reduction is a really important piece. And you can envision as we change the amount of oxygen that's in the ocean or in the atmosphere, we're going to be changing the nutrient budget. So we need to think about perhaps ways that we can evaluate the link between oxygenation and the nitrogen cycle, and how we begin to disentangle how oxygen and nitrogen have changed through time. And the way that I've primarily done this over the course of my career, and the way that a lot of other folks do it, is by using stable nitrogen isotopes. Primarily, I'm working with organic matter. That organic matter can come in a range of different kinds of forms. You can use whole rocks that have a lot of organic matter in them. You can use specific compounds, so like the nitrogen that is found in tetraperols of chlorophyll and chlorophyll derivatives, which have been found in rocks older than 1 billion years old, given special circumstances. You can use individual fossils. You can use the organic matter that gets preserved within the shells of bivalves. And this is really neat. I have a student who's working on this, where you can develop time series by serially sampling the bivalve and getting the sort of, perhaps a trophic type signal, but something that gives you a temporal change. And today, a lot of modern focused oceanographers and paleoceanographers are using the organic matter that gets preserved in foraminiferous. So there are a lot of different angles that you can get at. And, so sort of the beauty of the nitrogen cycle is that all of these transformations, that you could liken to a bowl of spaghetti or a bowl of noodles. There are a lot of interconnected parts. This is kind of confusing, right? There's a lot of passing back and forth of intermediates, but a lot of these have associated and predictable and measurable isotope fractionations that allow us to kind of get an estimate for sort of the balance of the major nitrogen cycle processes. And so we're going to kind of dive in here real quick and look at sort of the modern nitrogen cycle to get a sense of how we might dive into the deep time record. And so we can kind of think of the modern ocean as in balance between biological nitrogen fixation, so the primary source of nitrogen coming from cyanobacteria, and the process of nitrate reduction, which will be the primary loss mechanism for that nutrient nitrogen after it's oxidized. And this can happen in sediments, or it can happen in the water column. Sediments, important loss direction, but in terms of the isotope mass balance, this is a unidirectional process. The isotope fractionation that's associated with it, it runs to completion, so we don't see that signal. But where we do see it is in situations where we have anoxic waters juxtaposed next to oxic conditions. And along those interfaces is where we have nitrate reduction. And this has a very strong isotope signature. And so if we look at this diagram here and give you just sort of a sense of what's going on, here is the nitrogen isotope composition of, in this case, just dissolved inorganic nitrogen. And you can think of it as nitrate, like it is in the modern ocean. And then we have relative concentration here. So that's 1. So we would start out at this position. If nitrogen fixation occurs, this is adding nitrogen to the system. If we start out, in this case, at 5 per mil, nitrogen fixation looks a lot like the atmosphere in terms of its composition, which happens to be 0 per mil. It's what we use as our reference. That will decrease the nitrogen isotope values. And then if we have water column or incomplete nitrate reduction or ammonia oxidation, this has a very large fractionation. And we can see the decrease in the concentration accompanied by a significant isotope enrichment, right? So this is just pretty typical sort of kinetic fractionation associated with any sort of enzymatic reaction. And if we look at sort of modern systems in general, when we measure the isotope composition of organic matter in sediments, that matches the isotope composition of the nutrient source that these organisms were using. And so this kind of gives us the ability to venture back into time, although there is a lot of care that's needed and sort of assessing the preservation, since nitrogen does like to move around and it likes to be used by other organisms. So I'm going to step into one quick example. This is not my work, but it's a really nice way to look at what the nitrogen isotope record can give you, and also to some degree what it can't. This is a record from Eric Galbraith. It is looking at the last 50,000 years, and we're looking at the transition from the glacial to the most recent interglacial. And what we see in this particular case is at deglaciation, we see a significant increase in the nitrogen isotope values in places like the Mexican margin or the Somali margin. These are upwelling zones. They transition from oxidized systems to anoxic systems, at least in sort of intermediate waters. And this drives this isotope increase that's a signal for this water column nitrate reduction. Not everywhere is going to follow that track. So nitrogen, to some degree, is reflecting the local conditions. Its residence time is about 1,000 years. in the deep ocean. So we're not going to see this global secular nitrogen isotope curve. Now, what is thought to have happened in response to this nitrate reduction episode at the end of the last glacial is it depleted ocean nitrogen reservoir, and that tipped us back towards a situation where nitrogen fixing bacteria were able to make up that sort of removal, and we see a nitrogen isotope decrease as a dynamic response. So then that's sort of the framework. And at the point where I was a graduate student and getting into these kinds of things, starting off, this was the framework that as we understood. No one had really gone way back in deep time, though people were starting. And we had this sort of distribution of values. This is a histogram of modern core tops. They started about 0 per mil, which makes sense, right? That's the input value from the atmosphere from nitrogen fixing organisms, and the primary direction for the rest of those isotope values is increasing, and that's reflecting this nitrate reduction process. So if the modern is the way that we want to evaluate what's happening in the past, right, this was the scope, right? We have an oceanic anoxic event, dive into that, and you'd think like, oh, this is probably the signal that we're going to see. And this is what I honestly thought was going to happen. But obviously, I wouldn't be telling the story if that were true, because it wouldn't be very exciting. Okay, so oceanic anoxic events are episodes primarily confined to the Mesozoic, so from about 180 million years to about 65 million years ago. The Cretaceous is very well known for having these organic matter-rich sediments that are deposited sort of quasi-globally. If you go out into the Western interior Cretaceous sediments that you have, to the north and to the east of us, you will find all of these sorts of types of sediments, or in lots of places that you can recover using drill cores. And they do look like this generally. Some places it isn't quite as stark of a transition, but that's sort of the lithologic expression. And naturally, geologists are trying to understand how the world works. They're going to be drawn to this thing that's different and try to understand why it happened. There has been a lot of effort put into understanding these events, not just because we're curious, but because they're loaded with organic matter, and they are the bulk of some of the most active petroleum systems. So if we go back to an example in the Jurassic, one of the people who had been looking at these for a long time, a guy named Hugh Jenkins, went through one of these oceanic anoxic events and measured nitrogen isotopes, and sort of like a first stab at using this technique, found that the nitrogen isotope values in the core of this oceanic anoxic event increased. So it's like, oh, okay, that works. What really wasn't discussed is like, why are they so low? And I remember reading that paper and thinking like, why do they start out so low? So if we look at the oceanic anoxic record, not in sort of a stratigraphic sense, but just in a histogram here, what we find is that modern core tops, right, that histogram that I showed you before, do not overlap with oceanic anoxic events at all. So this on its own creates like, oh, well, there's an interesting story here, and we have to explain this. My first thought was this is some kind of alteration, or, like this can't be a real signal. This doesn't make sense. So we have to kind of get at this record and think about how this would work. And so if it is a real signal, the issue is you have to be able to explain why you have lots of nitrogen isotope values that are lower than the nitrogen fixation and member input. So if it's not around like, maybe 0 to -1 per mil, how do you shift the whole system lower, right? How do you get this non-overlapping distribution? So first step to this was, well, I was in an organic geochemistry lab, and I asked one of my committee members, well, what should I do? And she's like, well, you should look at chlorophyll derivatives and porphyrins. And so these sediments have extractable, basically you can think of it as chlorophyll. These are very well preserved in a lot of these sections, and you can find them, you can extract them, and they have nitrogen. And as it turns out, they confirmed that the nitrogen isotope values that we're measuring in total sediments were a real signal. There is a known biological offset between the organic matter of whole cell and its chlorophyll that typically is about 5 per mil, which is generally what we observed. So this suggested this is a real signal, and now we have to explain it. If we think about other sort of anoxic systems, we find similar types of things, low nitrogen isotope values during, sort of episodes of anoxia in the Devonian, so not just in the Mesozoic. And then if we also think about the Paleocene-Eocene thermal maximum, maybe not an oceanic anoxic event, but a big climate event that caused some areas to transition into a deoxygenated water column. Same deal. Nitrogen isotope values in this case look pretty much exactly like the model Cretaceous oceanic anoxic event. So how do we explain this? Basically, it's when you transition into an ocean that is anoxic at depth and has high nutrient concentrations and has a lot of ammonium in it. If we look at the additional sort of biomarker record, what we find is that we have phototrophic sulfide oxidation biomarkers preserved in these sediments. That means you had hydrogen sulfide well up into the photic zone. So this is perching this area of anoxic water well up into near surface waters where it can be mixed and entrained up into the surface. And what this does is, phytoplankton are really good at sucking up nutrients. And when they have ammonium, it's a party. And so there are the kinetics of that assimilation are very fast. And it ends up is that they outcompete the other organisms that like ammonia or ammonium, the nitrifiers. And there are sort of, matching sort of fractionations between these two. There is a negative isotope fractionation associated with the assimilation by the phytoplankton, and the nitrifiers kind of lose out. And there's this isotope partitioning between these two pools. This is sort of integrated over long periods of time, and this is what we think gives us these low nitrogen isotope values. You can run little box models, but it's also nice to kind of see it in a system that you can probe a little bit more clearly. And so I promise we will get to deeper time, but instead we're kind of venturing back to recent time. And we're going to use a modern anoxic basin. This is one of the great rift lakes of the East African Rift, Lake Kivu. It sits on the border between Rwanda and the Democratic Republic of Congo. So, you can work in sort of this side of the basin. And colleagues at Syracuse have been very active over the years in this basin. And I got the opportunity to go several years ago and collect some cores. And the deep water of this lake, it is a meromictic anoxic lake. It has chemocline that sits between about 70 and 100 meters. And it has the chemistry of this thing is really wild. And if you're interested in anoxic systems or lakes, we can talk about it more. But it has very high ammonium concentrations and very high methane concentrations, as well as CO2. It's one of these lakes that there's this sort of ongoing concern that there might be a limnic eruption. And it's possible that the sedimentary record records, if they're not actual proper limnic eruptions where you have eruption of CO2 and methane out into the surface, certainly these sort of overturning events that are often related to either earthquakes or hyperpycnal flows. There's arguments about what's driving that. And so this is a core image here. So these very, very finely laminated, extremely organic matter-rich sediments. Down here, we're talking like 35% organic carbon. Oh, I'm sorry, 20%. Still a lot. And even the background is really high, which is typical of these kinds of lakes. And they have these, intervals here, that the sapropel layers that have this very high organic carbon content compared to background. And what we see is this big negative kick in carbon isotopes. It's most likely this burp of isotopically depleted DIC or oxidized methane up into the surface. And we see this really interesting sort of depletion in nitrogen isotopes, which is this signal of nutrient abundance, and then this matching sort of rapid increase in nitrogen isotope values that is probably related to drawdown, so biological productivity, as well as nitrate reduction, and then some kind of relaxation. And so we're really excited about this, and then also very happy to see that increase, because this is not something that we can resolve at the kind of timescales in the rock record, because you're taking, from one of these Cretaceous cores, you're taking, you might take a meter and squeeze it down to 20 centimeters or something. So we're unable to sample at a resolution that yields us the ability to see, something that's happening on the scale of years, as opposed to maybe 1,000 years from an individual sample. So if we think about this process, we look at the data, and this is some modeling work from Ben Vegas, my former student, and kind of stereotype it, and you can build little models to think about where the fluxes are going. And it really gets down to this partitioning between phytoplankton and ammonia oxidation, which basically is shunting that right off to N2 and is lost from the system. You can generate these low nitrogen isotope values. And so if we then take this sort of idea and look at the ancient rock record, in this case, from the Mesozoic, Cenomanian, Turonian, 94 million years ago, we find, records that are sort of, varying back and forth. The isotope values are generally low. And, we see different behavior in different places. What we're doing here, though, is this is a stochastic sort of sampling of the Earth system, because, sedimentary basins are very dynamic. And, any one spot, like, okay, you build up a little pile, and then that pile moves over here, and it moves over here, and moves over here. So you're really just getting these like very intermittent snapshots when you go into the deep time record, whereas a lake is this nice, like varved, sort of terminal basin where all the sediment goes. And that's not how continental shelves or slopes work. So if we think about that and we expand this record, you can imagine, right, we have this sort of stereotyped variability that's going back and forth, and you squeeze it together. And that's kind of like what's happening perhaps during an oceanic anoxic event, where you have sort of repeated burps of ammonium as that chemocline is going up and down. Maybe this is driven by climate or some other external driver. And then if you want to go in and take that record, and this would be the whole record, and then this is what the geologic record is actually sampling, or maybe what you're sampling as you go up through the core, you can get this sort of just oscillation back and forth that gives you this, noisy, maybe, snapshot record. And so when we look at these deep time records, I am not making a big deal necessarily about, these little wiggles of a few per mil. We want to see something bigger. So this is just a nice way to think about this. And it was if you want to read a paper about nitrogen isotopes, Ben's recent paper I think it was in geology, is in my opinion the one to read. Okay, all right. So we have this sort of framework. We understand maybe how the modern works. We go back into deeper time. We have an idea of maybe how anoxic systems work. Is an OAE a model for what the Proterozoic looks like, where maybe you have an oxidized surface ocean and atmosphere, the degree to which we can argue about, but then anoxic deep waters. So would we expect to see this kind of isotope distribution? That's what I thought, but of course, nope. All right, just to reorient you, we're going to go in and we're going to dig into a couple of examples. First one is going to be in here right through the core of the Great Oxygenation event. And then another one that's going to be in here about 1.5 billion years ago in the Mesoproterozoic, and it's more pertinent to sort of issues of the evolution of eukaryotes. All right, so a lot of caveats here. This is the nitrogen isotope record through time. I did think earlier, I say that there's not really supposed to be a secular sort of one record that tells the whole story. range of variability, but we do have sort of external drivers that we might expect influence biogeochemistry broadly in the ocean, and obviously the nitrogen cycle is within that. When you go back into deep time, there are lots of additional considerations where we do have to worry about preservation. We don't have many rocks. A lot of the samples that I'm going to talk about in detail might come from one basin. And that's because we just are limited by the material that we have. So we're talking about up on continental crust, relatively shallow basins that have undergone a range of sort of geologic processes. Obviously, they haven't been totally subducted because they're up on this buoyant continental crust. But a lot of these old records, you can see this sort of light red color there, are highly metamorphosed. And people write these papers and think about this kind of stuff and still put these data in, and whether they're addressing something biogeochemical or if they're addressing something about tectonics, it's hard to put a lot of stock in some of these examples, in my opinion. But in general, the studies that have tried to understand how metamorphism affects these deeper time values, as long as there are not a lot of fluids involved, green schist facies, metamorphic rocks only generally drive isotope changes of a few per mil. And I said I wasn't going to make a big deal about 2 or 3 per mil. So that gives us at least a little bit of latitude. There still are some rocks that are, not super metamorphosed, but everything is rock, right? We obviously don't have mud. So let's look at this in a little bit more detail. We're going to start out and think about the 2.43 billion year old Deutschland and 2.33 Waihuka formations in South Africa. And so this is a really important sequence of rocks because it steps us right through this transitional period in Earth's atmosphere. So the key points here are that we have stratigraphy that is going right through this transition from the presence of no oxygen in the atmosphere, and this is we get this from the mass-independent fractionation of sulfur, so the sulfur isotope record, into a period where that mass-independent fractionation disappears. Now, what I'm not going to get into a lot of the details here is that there is a lot still that is not understood about the stratigraphy. And so in this particular spot here, you can see we have Botswana and South Africa. We have a series of cores that are taken by a range of entities. Agaron Institute was heavily involved in some of these. A lot of the data come from one spot, and then there's some new data that I'm going to show that come from at least a couple of different locations that are spatially separated. The degree to which these are correlated or not correlated, despite the fact that they have different ages, is outside of my area of expertise. And I know that if anybody here is into this stuff, will know that there are a lot of arguments about how these rocks fit together. Correlating rocks, even when you have relatively good radiometric ages, is not a straightforward process. So anyway, the key here really is that we have at least a better basinal spread to give us a sense of what's going on in this basin. Are there any differences in this isotope record? So here are some of the previous data. I'm hidden in the author's list of both of those. My main expertise, I guess you could say, beyond thinking about the nitrogen cycle and doing these kinds of things, is analyzing very small quantities of nitrogen and carbon. So when you have material that is either you have very little or it is very difficult to combust in large quantities when there's very little nitrogen, this is why people come to me. It ends up that most of these folks end up being my friends, which is fun. But in any case, some of these early studies, showed nitrogen isotope values that were rather enriched. So this on its own, you could interpret as the effect of oxygen on these biogeochemical systems that are driving isotope enrichment through the pairing of ammonium oxidation and nitrate reduction. Now, the signal being carried could be organisms using residual nitrate, or they might just be using that residual ammonium reservoir that's there. It kind of doesn't matter, but in any case, this is how these signals have been interpreted. And if we go in and look at this other section, these are new data from that were just submitted by Ben Vegas, I think maybe yesterday, that we produced in my lab over the course of the pandemic, which was fun. You can see this considerable increase to values as high as 20 per mil. And there aren't simple ways to explain this otherwise. There are some mechanisms out there that can, say volatilization of ammonia is one thing that's been kicked out there, though that requires, pHs that are quite high. And, maybe progressive depletion of nutrients is another way that you can do this. In any case, the isotope values, I think the most conservative way to explain these is the transition that we see in the atmosphere indicated by the loss of mass-independent fractionation of sulfur, which is oxygen in the atmosphere, which has to ultimately be coming from the ocean, right? From oxygenic photosynthesis. So in this case, if we then begin to bin these data and consider where and when and how this happens, if we go back and look at the Paleoarchean, right? So those are mostly cooked. In the Mesoarchean, there have been some suggestions from some folks that think that maybe there are hints of a redox cycle here, but for the most part, we have a distribution that centers around that 0 per mil. So this looks kind of like an oceanic anoxic event to some degree, but probably an ammonium ocean. So Neoarchean, we begin to see some of the hints of oxygen in a range of different types of proxies. And we begin to see a distribution that looks an awful lot like a lot more like the sort of modern distribution that's shown right up here. So certainly hints of O2, perhaps spatially variable, maybe confined primarily to the ocean. And then we have this transition here where we have loss of mass-independent fractionation of sulfur, clear evidence for oxygen in the atmosphere, and a distribution that continues to shift to the right that looks a lot more like what we see in the modern ocean, and that kind of continues onward. So, as you might imagine, this is a pretty data lean situation, and more data would be great. Better understanding of sort of post-depositional processes is something that I've started to work on, some experimental petrology types of experiments to basically simulate what happens to these rocks over geologic time and see if that can give us a little bit more context. There are lots of gaps, and it's not for lack of rocks. Like, we have plenty of rocks that are in these ages. It's just the time and the effort that it takes to make these kinds of analyses to start to fill this in. And obviously, good questions to dive into that. All right, so let's see. Next step here is we're going to bring in a slightly different flavor of my research that is a little bit more carbon focused, but has some implications for nitrogen. All right. So in the last few years, I've started working with a couple of colleagues, Susanna Porter at UC Santa Barbara and Phoebe Cohen at Williams College, and our postdoc that worked on this project, Hedda Agach. And for a long time, I've known Susanna and Phoebe for ages before I started working with them directly. I've been saying, like, hey, can we measure isotopes on these things called organic walled microfossils? Organic walled microfossils are, as you can imagine, they are complex organic matter that was produced by an algae or a metazoan, or all kinds of different organisms produce these. It's kind of similar to pollen in some degree, but they are one of the most important components of the Proterozoic fossil record. So before we have lots of visible life, if you look at the sort of remnants of a sediment after you macerate it, this is the kind of thing that you are going to find. So it's not that there's no fossils, they're just really small. And so they're primarily carbon containing. They're probably cells or resting cysts of primarily eukaryotic algae. There are some that are probably prokaryotic, but they have, in some cases, these really interesting sort of structures that, at least morphologically, at the sort of visual scale and at the micro scale, are similar to similar eukaryotes. A lot of them are probably polyphyletic, so it might be hard to say like, hey, this is a specific organism that's making this little sphere. It's a pretty common form in life, but in any case, the size and the microstructure suggests that they probably were eukaryotes and that they might be algae. But at this point, like, the morphology is the primary avenue that we've been able to use to say what we think these organisms were. So there's a lot about their ecology that needs to be resolved. And so what we started doing was measuring the carbon isotope composition of the individual microfossils. And so one of the things that we had to think about is, if we measure the isotope composition of these microfossils, what does it mean? Obviously, the hope is that it would encompass sort of the broad carbon isotope composition of that organism as it lived. It's possible that there are differences between sort of interior components of the cell and the outside part that gets preserved. It might reflect where they lived. It might reflect the broader carbon cycle. There were a lot of things that we really didn't understand when we started this, because there have been some attempts to do this using SIMS, but not at a broad scale and not at sort of high numbers, which is what we're trying to do. And so the Mesoproterozoic Roper group is in Australia. It is a shallow epicratonic basin. Like all of our Proterozoic basins, we're basically sampling the shallow ocean. It has wonderfully preserved microfossils and a complementary organic geochemical record is something that we just don't find in rocks that are 1.4 billion years old. So we have records of sort of protoforms of sterols or steranes that suggest that eukaryotic organisms were active in this environment. And so it's an important place for us, for folks who are trying to understand sort of the evolutionary pathway of eukaryotes. All right, so this was one of our first efforts in diving into making analyses of individual organisms. So what we do is we would take this single microfossil off of a slide, pick it with a little, sort of picking needle, put it into an elemental analyzer cup that's really small, and put it into a special elemental analyzer that's coupled to all kinds of traps and things, and capture and concentrate that carbon dioxide because it's down sort of at the level of the amount of carbon that you might need to do sort of compound specific carbon isotope analysis. So in that kind of nanomole region. So really small amounts of carbon, and they're kind of difficult to deal with, but kind of fun to get after this and try to make some sense of it. So in this particular section, we're just going after kind of the simplest forms. And what you can see here, we have core depth. We have sort of focused on the upper part of this section, where if you look at sort of the redox data here, this is molybdenum concentration. We have a transition from a euxinic basin to a more oxidized system. Probably a change in depth associated with this. And below this, we don't have any microfossils, but up here through this transition from euxinic to oxidized, we see A general sort of decrease in the values, and then also especially a lot of spread. And when we first started analyzing these, I was trying to make sense of why we have this amazing range, in this case, 10, 12 per mil in one single sample for these individuals. And if you think about an organism that lives for a couple of weeks, or maybe a month, maybe a couple days, and this sample is encompassing thousands of years, perhaps, of time, we have a significant time averaging effect, and then just sort of the individual differences of one organism living in a particular place that might result in this sort of breadth of carbon isotope values. The one thing that is nice is that it is well within the range of isotope values that we would expect for conventional carbon fixation. So the idea is that this fits that these organisms are probably phototrophic. They're probably eukaryotic algae. And so, we did pair this up with at least a little bit of nitrogen isotope data in this basin that matches other periods in the Proterozoic, and it's generally consistent with the presence of a redox active nitrogen cycle. And so what we think that we're seeing here over the course of the upper part of this section, go ahead and, can I erase all of this? Erase pen. There you go. Look at that. Is a transition from organisms that are living primarily at the surface and held into an oxygenated surface zone, and then we're able to move into the rest of that water column. And sort of the way that we're interpreting this at this point is that in the anoxic period, we have probably what is a strong carbon isotope gradient that's related to sort of productivity exporting the isotopically depleted carbon down to depth, and our microfossils living right at the surface, and a relatively oxidized water column based on those redox proxies, and these organisms probably inhabiting a much wider range. So they're sampling sort of a carbon isotope gradient that is much broader, and so not held up at that surface. And so basically, what we get out of this, it doesn't seem, I don't know, I mean, maybe it seems exciting, but it's exciting to me, I don't know about to you, is that we have never been able to make these kinds of analyses before to kind of pin where we think that these organisms were living based on something other than their morphology, and then association with other redox proxies. And so this at least gives us a hint that they are changing their ecology, in this case, probably based on the redox state. And so one of the ideas that's been out there for a while, and I think we have probably one of your faculty members here responsible for sort of spreading that out. So I know that Ariel, you put this idea out there a while ago, and the rest of us have been chasing those ideas for the last many years. And that as the Earth transitioned from a low oxygen to a higher oxygen space, the partitioning of where nutrients were might have driven organisms to live in different places. So if eukaryotes favored surface waters, this might have been a place that didn't have a lot of nutrient available. And so they might have had to find new and unique ways to access the nitrogen and the phosphorus and the iron that they needed. And one of the things that we know that modern eukaryotic algae are really good at is they're good at sucking up nitrogen, especially nitrate in surface waters. And so this might be sort of one of the things that we can get at with these kinds of analyses. We don't have nitrogen isotope analyses on these. Hopefully we can do that at some point. But it's consistent with the possibility that they were nitrate specialists, which is an interesting piece in connecting the evolutionary history of eukaryotes between sort of the early common ancestors to the stem groups to the crown groups that we now have today, that we know that some of them really favor and do well in those types of environments. All right, so there are lots of other places where we're active in kind of attacking these types of problems, using microfossils, using nitrogen, other parts of the mid-Proterozoic we're getting after using the same kinds of techniques. I have a bunch of other things if you're interested in, but I hope that you enjoyed that, and hopefully you have some questions. So thanks for having me. Right up.
Hi. I have to push a button. I can't remember. No. How do you see the impacts of or the potential role of that or other forms of alteration in these isotopic signatures, or is that too big a question?
So no, it's not too big of a question. It's a real question. Obviously, the thermal maturity of the rock is the primary control. There have been at least some reasonable studies on metamorphic rocks that suggest that, if there aren't a lot of fluids involved and the metamorphic grade is sub-green schist, that the isotope difference is relatively small. We also we extract the organic matter and measure the organic matter. We measure the mineral phase, look at the concentrations, and generally what you should see is not a huge offset between, say, nitrogen that might be, in the whole sediment and the organic matter. So nitrogen can insert itself into clays, and this is a common process that happens, during metamorphism. So we have to make those kinds of analyses to be able to assess that possibility, right? I mean, look at the minerals. There are a range of different ways that we can get after it. So it's not perfect, but yeah, and like every single paper somewhere it needs to have it, right? And there has been a move. I don't know, there are a couple of different camps out there that think that like the whole rock is the better representation of that, because the mineral nitrogen that's there probably is derived from the degradation of ambient organic matter. And we see that in more modern systems where if you measure the mineral nitrogen in Cretaceous black shales and the organic matter, they're isotopically almost exact. So the maturation process and metamorphic process is probably driving some difference, probably as a part of the mineralogical change.
We have a question online. Did our atmosphere always have nitrogen as a dominant component?
Yes, and this is a really interesting and evolving story. I think that a long time ago I would have said, like, oh, nitrogen content has always been the same. It turns out that maybe that's not the case. So the amount of nitrogen in the atmosphere has possibly changed a lot. It's probably still been the most abundant gas. And it has some really interesting effects in how it interacts with the other gases. So there is a growing and rich literature on sort of early atmospheric composition, and what you think is, oh yeah, boring nitrogen gas it's a really cool story. So I would Colin Goldblatt and Eva Stoiken and a few others have dug into this, and I'd urge you to check out their work.
I guess this is sort of in the same vein. How would, like, in theory, would the nitrogen isotope composition of the atmosphere be changing over time, and how would that affect like measurements you're taking with the fixation?
Yeah, right. So that's another idea that we've kicked around. It's possible the time period that we're talking about with big swings in the amount of nitrogen in the atmosphere are really early, not in the time periods where we're really focused on. So I think the idea is that it's a pretty stable reservoir isotopically, but it's certainly something that you'd think you'd have to consider as to whether there is change. So you could model it to see how a small change would then propagate through our records. So, and I'm pretty sure that Colin and Ava have thought a little bit about that as well. So it's a great question.
Okay, I have going here. I did my second project in grad school on aquatarks and their isotope composition, so it brought me back. So I was wondering, what are the processes that would drive an algae to have that wide of a range in carbon isotope composition?
That's a great question. I would think, I mean, we're sort of pulling on if you look at sort of modern organisms, and there's been some really cool sort of like Sims types of analyses where they look at sort of the metabolic rates, thinking mostly from a nitrogen space. But the same thing in terms of like total carbon fixation. Maybe they're not phototrophs and they're mixotrophs and they're just eating different things. Like, so there are a bunch of different ways that I think that you could explain it, just in terms of also like, the time period involved in that single sample, the amount of carbon isotope variability that you might have in dissolved inorganic nitrogen or dissolved inorganic carbon, or the composition of the atmosphere, or whatever carbon substrate they're using, can yield us that breadth. That's sort of the idea.
So I'm curious whether the GOE led to a net increase of atmospheric pressure, or if there was some means that, was it always 1 bar, and did it start out at 80%, 0.8 bar, and then it jumped up to 1? Yeah, what did that look like?
So not discussed here is, there are different models for the total content of oxygen in the atmosphere, and one of the ideas that has been out there for a little while is that the initial pulse and increase in oxygen drove an overshoot, that the amount of oxygen in the atmosphere might have gone up to near modern levels. And in part because there's no like clear negative feedback. Like today, if you have 35 or 36% oxygen in the atmosphere, like things start burning and they don't stop burning until there's less oxygen. So there is the idea that there's this upper threshold of what oxygen can be in a, plant-rich terrestrial surface. But back then there might not have been. And definitely if we think about other periods in the past where we're pretty confident that oxygen concentrations got really high, like in the middle Carboniferous, we're growing this new biosphere on what was prior to that a relatively, plant-free surface and burying a whole bunch of coal, right? You're pumping a lot of oxygen into the atmosphere, and that the atmospheric pressure actually did go up. So yeah, it's additive.
Hi. So I'm really curious about the 20 per mil, like delta 15N excursion that you see in the GOE. So more specifically on the lack of sort of a shift in the delta carbon 13, because I would have thought that, a shift in the population of like microbes like dominated by photosynthesis would show up. And you kind of alluded to something analogous with the Mesoproterozoic, right? So I'm curious as to like how you're thinking about this particular record of like organic carbon at the time.
Yeah, so at least in here, I mean, I know that they're focusing on that little decrease. There's this decrease in another section, though it obviously doesn't continue to go up there. It's hard to see here, but if you zoom in on that one, and then that one there, the interpretation for those decreases is, you're burping a whole bunch of oxygen into the atmosphere, and you're oxidizing either methane that was in the atmosphere, or maybe methane that was in the ocean, or you're oxidizing organic matter that's present in rocks and that's weathering, and that you're seeing a decrease. Now, as far as the thinking about farther up that core, I guess I don't know why you would have to have a compensatory change in the carbon isotopes compared to a change in the nitrogen isotopes. There are lots of instances where you can have nitrogen doing this and carbon goes like that. Though, like, in an OAE, they obviously, they're all moving together. So does that answer your question, or is that a dodge?
Maybe. So when you showed the sort of modern Lake Kiva nitrogen stuff, and you showed that very rapid weeks to months kind of change in nitrogen, and then the model with the nitrogen doing like that in the low resolution sampling, the biogeochemist in me was very happy to see that, yes, nitrogen could change really fast like that. Slightly disturbing from a sediment geochemistry standpoint. So my question is really trying to pull these two things together. Could the variation in your microfossil carbon be something like that high resolution shift due to biogeochemistry?
Sure.
And how do you unpack that?
I mean, maybe you could look at it as that isotopic breadth on its own is something interesting that you could maybe interpret in that context. Can I absolutely explain it? No, would I like the more paleo-focused biogeochemist that I am, I probably should go and start analyzing single diatoms for their carbon isotope composition or other modern algae. That would be a good place to start. And it's probably something that a master's student could do.
Say.
Oh, okay. Probably also what maybe the other question about the range of variability was hoping to hear. So thanks for helping me.
Well, if not, let's thank Chris for a great talk.